Dr. Duffy is an associate professor at the University of Michigan in their Ecology and Evolutionary Biology department, where her research focuses mainly on the evolutionary and community ecology of infectious diseases, as we'll hear about today. Prior to her appointment in Michigan, she was at Georgia Tech, and she has a real amazing record of academic scholarship. I encourage you to Google her. It's quite impressive. One of her most notable accomplishments was the award of Presidential Early Career Award for Scientists and Engineers, or PCASE. This is the highest honor human faculty member can win in our country. Aside from these notable academic accomplishments, she's a strong advocate of outreach and engagement in sciences. And last year was on the lead stage at the March for Science in Washington, D.C. And she's active on Twitter, which is how I came to follow her at Duffy MA. And with that, let's start your session.
Thank you. And thank you all for coming, some of you for second day in a row, so I'm glad you're back for more Daphnia disease stuff. So today I'll be talking about my slow transition into ecosystem ecology. I definitely don't view myself as an ecosystem ecologist, but it's an area we've been doing more and more work in. But as I did last night, I want to start out by acknowledging the people who are co-authors on the work that I'll be talking about today. So I'll be talking about a few different projects today, so there's a longer list of co-authors. But notably, and I'll highlight especially important people as data appear along the way, but notably everyone who's underlined is an undergrad who worked in the lab and who will be a co-author on publications resulting from the work. So as I said yesterday, one of the things that I really love about our system is that it's very tractable for undergraduate student projects. And so we really like having undergrads in the lab. We have 11 this semester, which is probably pushing it, but we really like having undergrads around and involved in the research. And then again, similar to yesterday, I want to also start with a territorial acknowledgement, acknowledging that this land was originally the home of Native peoples. And so I talked a little bit about these tribes yesterday, and so I'll try to highlight different things today. So one thing that I've found really interesting about doing territorial acknowledgements is reading about different tribes. And so one thing that stood out to me when I looked at the Shoshone-Bannock tribe's website is they have a climate change assessment. It's pretty prominently featured on their site. And so it talks, if you watch this video that I've screen capped part of here, it talks about how they chose to live in the Snake River area because of the water, which is extremely important to them. And it also talks about the importance of sagebrush, which is used for medicinal purposes. And it talks about the changes to the water and vegetation that have occurred with climate change and how they're working to prepare for this changing climate. And then I talked yesterday about Sacagawea, who's a well-known Eastern Shoshone woman. And so today I'll note Chief Washakie, who negotiated the original reservation with the U.S., which was actually 44 million square acres, so much bigger than the current one. Okay, so I like to begin with an observation that some people find depressing, some people find interesting, which is that parasites are everywhere. So parasites affect humans, they affect organisms we care about for multiple reasons. And so this includes organisms of conservation concern. I mentioned briefly last night that there's a fungal disease that's been decimating bat populations, especially in the eastern U.S. That's white nose syndrome. Humans, again, are unfortunately the subject or the hosts for many infectious diseases. Zika is one that captured a lot of attention recently in the media. And then diseases also threaten organisms of economic importance, and in the case of coffee rust, of academic productivity importance. And so we care a lot about infectious diseases. And again, in part just because parasites are so common, it's really important to understand their effects on host populations and communities and ecosystems, as well as to understand what factors control the size and the timing of infectious diseases. And so this is something that people have worked on for a long time. People have worked on host-parasite interactions and the ecology of host-parasite interactions. But they've tended to do that looking at a single host species and a single parasite species in isolation. And in reality, of course, that single host-parasite interaction is embedded in a much larger food web. And this is still a ridiculously simplified food web, right? But there's still much more going on here. And so we know now that these other food web members are really important for disease. And we're also starting to understand now how diseases themselves can influence food webs. And this is true in the particular system that I work on. And so this was a convenient cartoon that was actually made by someone else. But it's also true more generally. We're starting to understand more about these links between diseases and food webs and diseases. And so if we consider links between ecosystems and disease, if we consider the ways that ecosystems can influence disease, right, so the arrow going in that direction, we know for these, this trematode parasite, this Riboroia trematode, that eutrophic systems that have higher productivity levels, have more snails, and snails are the intermediate host for this, and then you get more disease, so more deformed frogs in a population as you have increasing nutrient levels and increasing primary productivity. And then if we take the arrow and we move it back in the other direction and we look for effects of disease on ecosystems, I don't think I've ever seen a more classic trophic cascade figure than this one. So this is from a relatively new paper looking at sea star wasting disease, which has been spreading on the West Coast. It's probably caused by a virus. And so this disease has come and really swept through many areas on the Pacific West Coast. And so if you look at this figure here, if you look at these sunflower stars that are one of the hosts for it. So before the outbreak is the dark bar. They were pretty common, and then after the outbreak, they're quite rare. And then they feed on these sea urchins, which become much more common after the outbreak. And then if you look at the kelp, the primary producers in this system, they become much less common after the outbreak. So and it's more than just, there are multiple things going on here, but it's clear that the disease is influencing the ecosystem in this case. And you can imagine then that given these changes, especially in the kelp, that you would have changes in primary productivity and nutrient cycling, and it would change a lot of ecosystem properties as well. So we have some examples, but it's still pretty early days in terms of understanding these links between ecosystems and disease. And my talk today will be in two parts. So in the first, I'll look for an effect of ecosystems on disease. And I'll talk about lake browning, which is something that hasn't received a whole lot of attention, but it's a component of global climate change. And I'll ask whether a browner world might be a sicker world, and look for influences of water clarity on patterns of disease. And then in the second part, I'll look for influences of disease on ecosystems and tell a story about how disease can actually increase host density by altering the primary productivity in a system. Okay, so starting with this first part here, this is work that's part of the PhD dissertation work of my graduate student, Clara Shaw. And then it's done in close collaboration with Spencer Hall, who's a long-term collaborator of mine at Indiana University. And then with Aaron Overholt and Craig Williamson at Miami of Ohio. And they're the ones who know all about characterizing light environments in lakes. Okay, so I mentioned before how it's important to consider host-parasite interactions as being embedded in a larger food web. And what I should add to that is it's also important to consider the ecosystem context of that food web, so the larger abiotic context in which that food web is embedded. And so the key difference that I'll be focusing on for this first half of my talk is that some lakes are clear and some lakes are brown. That may have been too subtle, right? Clear, brown, right? So lakes differ in their water clarity, and they differ in this naturally, so there's natural variation in the dissolved organic matter that ends up in lakes, but this is also something that's changing with global climate change. And so for this part of the talk, I'll be asking about whether light drives Daphnia parasite dynamics. And then more specifically, asking about whether two common Daphnia parasites, the same two that I talked about yesterday, are differentially sensitive to light, and whether those effects might be important then for epidemic dynamics in lakes. Okay, so I mentioned before that lakes are getting browner as part of global climate change, but this is an underappreciated aspect of global climate change. And one key driver of this is that with climate change, you get more storm events and more intense storm events, and those bring more organic matter into lakes. And you can think of this, or when you make tea, you're dissolving organic matter in water. So with climate change, we're making stronger lake tea. We're dissolving more organic matter in our lakes, and we're making them browner. And so this has the potential to have a lot of consequences. Solar radiation is obviously really important for powering the base of food webs, right? All the primary producers you can see here and the ones you can't see below the surface are relying on that solar radiation. But it also has the potential to be damaging. So if you were out sampling this lake on this beautiful sunny day in the summer and you didn't wear a hat and sunscreen, you might end up looking like that by the end of the day. And it isn't just aquatic ecologists who are at risk of being damaged by the sun. Other organisms can be damaged by the sun as well. And we've seen this in earlier work that we've done in this system, that the fungal parasite we work on is pretty sensitive to light. And that's both to UV radiation, but also to photosynthetically active radiation. It's actually kind of a wimp when it comes to being exposed to light. Okay, so I mentioned before that we know lakes differ in terms of the amount of organic matter they have, and therefore in terms of how much solar radiation they receive. So some lakes are naturally darker than others. They naturally have more dissolved organic matter, and that dissolved organic matter absorbs especially high energy wavelengths. And so light doesn't penetrate as deep into a lake if it has higher levels of organic matter. And so what we were interested in is whether this organic matter is basically serving as sunscreen for some parasites, and maybe differentially serving as sunscreen. And throughout my talk today, I'll use this clear glass of water that I would drink and that really nasty glass of water that I would not drink to indicate these ends of the spectrum if you're not used to thinking about dissolved organic matter. Okay, and then, so there's definitely spatial variation across lakes in terms of how brown they are, but there's also, and therefore in terms of how much solar radiation they receive, but there's also seasonal variation in how much solar radiation we receive. So parasite epidemics tend to take place in July through November in these lakes, and during that time, light changes from being pretty high intensity to much lower intensity. We wear much less sunscreen when we're out sampling in November for multiple reasons compared to when we're out sampling in July. And so I'll use our nice summer beach umbrella to indicate when we're in the summer and high solar radiation, and then our beautiful maple leaf for when we're in the fall with less radiation. Okay, so then just to remind you of these two questions that we were going to address, we're interested generally in whether light is differentially impacting these two parasites, and then whether that has important effects for epidemic dynamics. And so we did this with the same study system that I talked about last night as sort of the workhorse study system in the lab, where the host is Daphnia dentifera, so it's a key dominant species in these stratified lakes in the upper Midwest. And so they're a key link in these aquatic food webs. They eat the phytoplankton and fish eat them. So they're really ecologically important in these lakes. And they host a wide variety of parasites. Two of the most common ones are shown up here. So this fungal parasite, Mentionichellia bicuspidata, which makes these needle-shaped spores. And then the bacterium, Pasteurella ramosa, which makes these spores that look like sunny-side-up eggs. And I mentioned before that we already know one of these parasites is sensitive to light, and that was the fungal parasite. Okay, so then for this experiment, what Clara did is she took parasite spores. She took infected Daphnia. She ground them up to make these parasite spore slurries. We tend to say in the methods that we gently smash them, which actually makes no sense, but that's just sort of become how we describe it. So she gently smashed infected hosts, and she made parasite spore slurries, and then she put them in these quartz vials. And then she incubated the quartz vials in lakes for 5 days. And some of the vials had the fungal parasite, and some of the vials had the bacterial parasite. None of them had both. I'm not leaving out a treatment today. And then she had ones that were light, and she had ones that were dark, where she wrapped dark plastic around them so that light couldn't get through the vial. And then she incubated them at 2 depths. It's kind of hard to see in this picture, but you can just barely make out up here the half-meter depth. So they're on, we call them trees, these sort of things with lots of branches that she attaches all the vials to. And then you can actually also just kind of make out the 2-meter depth one there if you squint a little bit. So she had 2 depths where we would expect then higher solar radiation at half-meter than at 2 meters. And then she did this in 4 to 5 lakes that ranged from being pretty clear to being relatively dark. None of our lakes are super brown, but they get relatively dark. The reason it changed from 5 lakes to 4 is we lost access to one of our long-term study lakes in the middle of this experiment, which was quite disappointing. And then she did this 3 times. She did it in July, in August, and then the last one actually straddled October into November. And then this picture at the bottom shows, so this is, you can see the little floats at the top of one of the rafts. That's those floats there, and then they have these other floats anchoring them in the lake. Okay, and then, so after she did this incubation where they were out in the lake for 5 days, she went back out, collected all of those quartz vials, brought them back into the lab, exposed Daphnia to the spores from those vials, and then kept them going in the lab until she could tell whether they were infected or not. Okay, and so I'm going to eventually show you a larger graph, but I'm going to start with this one just to walk you through it a little bit. So this is the data for the bacterial parasite for 1 month, just for the first month of July. So there were the ones that were in the dark, where remember that quartz vial was wrapped with dark plastic. That's shown in the dark blue. And then, sorry if I'm blocking someone. And then the ones in the light are shown in this lighter blue. And then there was the shallower depth, the half-meter, and the deeper depth, the 2 meters. And then this is proportion infected again, where it goes from none getting infected to all getting infected. And so with this bacterial parasite, you can see, especially here at a half-meter, the ones that were incubated in the dark did just great. Most of the hosts got infected. The parasite was very happy, whereas they were basically all killed by this incubation at a half-meter when they were exposed to the light. And then you see a difference at 2 meters, but it's less strong. And again, that's what we would expect because there's less radiation reaching 2 meters. Okay, so now if we look across time for this bacterial parasite, going from our nice summer days to our cooler, darker fall days, you can see there was this really dramatic effect in July, and by October, there's no difference between the light and the dark treatments or the shallower and the deeper depths. So the bacterial parasite is clearly sensitive to light, but the effect of light on it, as the light wanes over the season, it stops having an effect on the parasite out in our natural populations. Okay, so now if we add on the fungal parasite, so the initial spore dose she used was too low. So July was just kind of a dud. But fortunately, so then for August and October, she used a somewhat higher spore dose and had more infections. And in October, she still saw really dramatic effects on the parasite, on the fungal parasite. So pretty, decent infections in the dark treatment for a half-meter and 2 meters, but no infections for the ones that were exposed to the light. And so this comparison, so the reason it's fortunate that we got this worked out by October is this comparison here lets us say that the fungal parasite is more sensitive to light than the bacterial parasite is, because when these light levels were low in October, the fungal parasite was still impacted and the bacterial parasite wasn't. So that tells us that it's more sensitive. Okay, so we can say now that yes, these 2 parasites are differentially sensitive to light, and so then we wanted to know whether those differences were important for epidemic dynamics that were happening in lakes. And since we found that the fungus was more sensitive to light, and it continued to be sensitive to light well into the season, we expected that epidemics of the fungus might start later, right? So as it might need light to drop off even more than the bacterial parasite does before it could really get going. So we thought that we might see the bacterial epidemics take off sooner than the fungal epidemics. And then we thought that if we looked across lakes that differed in their clarity, that these darker lakes might have larger outbreaks of both parasites than the clearer lakes. And that would be because the darkness of these lakes serves as sunscreen for the parasites. So they might be able to live and infect better in these darker lakes. Okay, and so looking at just what's going on with when the epidemics start. So Clara used data from 3 different years, so 2014, 15, and 16, and this was based on a long-term sampling that's led by my collaborator, Spencer Hall. And so the fungal parasite is shown here in the light blue, and the bacterial parasite is shown in this orange color. And in all 3 years, the epidemics of the bacterial parasite started earlier, and overall there's a significant difference between these 2 parasites in terms of when the epidemics start. This is for all of the lakes that had epidemics of at least one of the parasites in a given lake year. We also did an analysis with only the lakes that had epidemics of both of them in case there was some within lake factor that was important. And even if you use that narrower data set, you still see the same pattern. And then we did find that bacterial epidemics become larger in darker lakes. So we have this UV transparency index here, which goes from clear lakes over on the left to darker lakes over on the right. And then epidemic size, what we do is we take the epidemic curve that we get, so the infection prevalence over time, and we integrate under that, and that gives us an integrated measure of the size of the epidemic. And so you can see that there's this positive relationship in all the years. And actually what's more interesting is that, so we have this criterion for what we count as an epidemic that's something based on work I did as a grad student, and we've used it ever since then. And so actually the only lakes that actually hit this threshold where we would actually consider it an epidemic, tended to be the darker lakes. So actually, in the clearer lakes, the bacterium just can't get going. It's just too killed off by the light, it seems, to even make it to the point of what we would call an epidemic. Surprisingly, we did not see that fungal epidemics became larger in darker lakes. The relationships were not significant for the fungal parasite, whereas we'd expected, if anything, that we might see stronger effects because the fungal parasite was more sensitive to light. Okay, so if we come back to these questions that Clara had, so are the 2 common Daphnia parasites differentially sensitive to light? Yes, they are both sensitive, but the fungal parasite is more sensitive than the bacterial one. And then asking whether those differences might be important for epidemic dynamics, it does seem, especially for the bacterium, like light is structuring the timing and the size of the epidemics that we see. So the bacterial epidemics start earlier than the fungal epidemics, and the bacterial epidemics became larger in darker lakes, though for unknown reason, we didn't see that pattern with the fungal epidemics. So remember that lakes in several parts of the world are getting browner, and so this is data from one lake, Lake Giles in Pennsylvania, which has been a long-term study site of our collaborator, Craig Williamson. And so this is looking across time. This is looking at the percent UV depth. It's a classic limnology graph where you have 0 at the top, so we tend to flip our axes. And you can see that these lakes are getting browner. The depth of UV penetration is getting much shallower over time. And so our data suggests that this aspect of global change might be making it easier for parasites and making it more likely that we'll have parasite outbreaks in these lakes. And this is something, there's a concern that this might apply for aquatic diseases more generally. So we don't have data of the sort that we have on these Daphnia parasites for very many diseases. But we know that many pathogens are sensitive to UV radiation, and we use UV light to sterilize water and to sterilize surfaces, right? And so if one aspect of global change is making it so that UV doesn't penetrate as far, we're going to lose that natural sterilization, the sort of ecosystem service that most of us aren't aware is happening out there. That might be impacted by global change. Okay, so this first part has been about how this ecosystem property of water clarity and DOM might influence parasites. And it relates to work that's being done by one of the postdocs in my lab, Nina Whale, and an undergrad who's doing her honors thesis on this, McKenna Turrell. And so this is a different parasite that I haven't mentioned yet. It's one it's never super common in our lakes, but it's always around at some level. So but it doesn't have these really big epidemic sizes, but it's beautiful. And so we've always noticed this. And it's a parasite that was actually originally described in the 1880s by Ilya Mechnikov, and he was drawn to it because of this really brilliant red color that it has. And so we in my lab have tended to refer to it as scarlet for fairly obvious reasons. If you look at the top there, I should probably take video because it also sort of glitters as the cells circulate in the bottom. It's a really, really striking parasite. But we noticed over the years that we had something that had the same distribution in the body and that same glittery appearance, but they were white. And we started calling it white scarlet, which doesn't make any sense, except it sort of made sense given the work we were doing in the lab. And what Nina and McKenna have started to work out is that this actually seems to be driven by the light environment. And so I was a little suspicious when Nina first started proposing this. And then she did this experiment, and I was like, okay, you're right, this is super cool. So what she did in this experiment is she either infected some animals. So Nina has done a ton of work to get this set up as a lab system where we can infect in the lab. This is something I've wanted to do since I was a grad student, and I couldn't work it out, and Nina did, and it's super cool. And so she and McKenna exposed some animals in the lab and then put some animals in the light and some animals in the dark. And these three super bright ones are typical of the infections that we see when these animals are grown in the light in our sort of normal lab conditions. Whereas the ones at the bottom were all grown in the dark. And so you see much more variation in color. You do get some that are fairly red, though they never get quite as red as the ones in the light. And then you get some ones that fall into this white scarlet category where they have the opacity in their body, but they don't have the red color. So this is pretty fun, and now we're starting to look at how light environment might affect this phenotypic plasticity in this parasite, which again is one of these never saw this area of research happening, but it's pretty fun. And something that's interesting, and the thing that really got Nina thinking about this, so she's really good at thinking really broadly, and she'd read that Myxococcus xanthus, which is a relatively close relative of the bacterium that causes this, that it produces carotenoids in response to exposure to light. And we know that this is a carotenoid based on work that was done a while back and that we've been repeating. And they produce this carotenoid because it has a photoprotective effect. And so she's now really taking the Myxococcus literature and applying it to this new system, which has been a lot of fun. Okay. So that's one example of how ecosystems might influence disease. And so let's move to the second part to think about how diseases might influence ecosystems. Okay, so we thought that disease might, or generally if you think about this, disease might influence ecosystems by direct density-mediated effects. So I mentioned Daphne are really important grazers in these systems. And so maybe if you have a big disease outbreak and it kills off a lot of the hosts, then you might get an algal bloom. But there also could be trait-mediated indirect effects, and this is something that's emerged over the past, 20 to 30 years or so, is something that's studied a lot in ecology. So including my colleague Earl Werner has led a lot of this work, where we now realize that there are a lot of impacts that are not caused by things other than organisms dying, right? There are other impacts that can be really important. And so we thought, well, maybe if the diseases have an impact on Daphnia feeding rate, maybe that could also influence primary productivity. Maybe that could drive an algal bloom. And so where we're gonna go with this is that the effects of disease on an ecosystem might end up then feeding back to a host population. And so this is work that was part of the PhD dissertation of Rachel Penzikowski, who was my first grad student, and she's now in her first year of a faculty position at Washington University in St. Louis. And then this was done in very close collaboration with my longtime collaborator, Spencer Hall at Indiana. Rachel actually went to Indiana to do a lot of this work. The field work is all based out of Spencer's lab. Okay, so for this part of the talk, it's really important to talk about feeding rate. So feeding rate is the super important trait in our system. So feeding rate ends up influencing the amount of algae that, so phytoplankton generally, that hosts consume. That seems obvious, right? It's the feeding rate. But because the Daphnia become infected when they accidentally ingest parasite spores while they're feeding, the feeding rate then also influences their exposure to the parasite. And then because once these infected hosts, as they take in food, that's what the parasite is stealing to use for its own growth. So the ones that take in more food then can grow up more parasites. So feeding rate is super important because it influences algal resource consumption, but also exposure to parasite spores, and then the number of spores that are released from an infected host. Okay, and this is a slightly complicated story, so I'm going to tell you what I'm going to tell you, and then I'll tell you, and then I'll tell you what I told you. Okay, and so in the first part, Rachel used a lab experiment to show that parasites reduce feeding rates of infected hosts. And then in the second part, she took those the things she found in the lab, and she used that to parameterize models to see what effects we should expect of those effects on feeding rate. And then that predicted some pretty exciting things that we hadn't necessarily expected. And so then we went and looked at our field data to see if we could find evidence of that in our natural populations. And the take-home message will be that parasite epidemics can actually increase host density by depressing feeding rate on algal resources. And so this is something that's known as a hydra effect. And because I work on small aquatic organisms, people sometimes think I mean the small aquatic organism hydra when I talk about this. This is the mythological hydra. So where you cut off one head and two grow in return, right? So where you have an increase in mortality that actually leads to an increase in density. That's known as a hydra effect. Okay, so for the first part of this, Rachel did a lot of work to work out these feeding rate assays, and this is now something we do all the time in the lab, but Rachel did a lot of work to get them, the methods worked out for this. And Rachel does things in a big way. So she used 3 different genotypes and then a whole range of ages and stages of infection to see how these traits change with age and with infection. Okay, and so if we start just with the uninfected animals, so she had the 3 genotypes, so each of the panels is a different genotype. She had them across, again, a really impressive range of ages. And then the feeding rate is shown on the x-axis here. And this is showing just the data for the uninfected hosts, and you can see that their feeding rate increases and generally saturates as they get older and bigger. If we add on the infected hosts, which are shown with these filled circles, you can see that infection depresses the feeding rate really substantially, right? So these infected hosts are eating much less than their uninfected counterparts. And again, this is the same genotype and the same age. So we would expect them otherwise to be the same. And so if I add on the stats, there are significant effects here, but it gets a little overwhelming to have all that on the slide at once. And so this is something where we did this work thinking that maybe there would be an effect there. And part of why we thought there might be an effect there is this picture that I showed last night. So this is a scanning electron micrograph where I've sliced through the carapace of a Daphnia, and we're looking into her body cavity. And by the end of infection, it's just completely packed with these fungal spores, which are like needles. And I could imagine if I was filled with a whole lot of needles, I might not feel like eating very much. So we weren't surprised that there was an effect, but we were surprised by how early the effect started. So what I didn't mention before is that we only exposed these animals to the parasite when they were 6 days old. And so by 2 to 4 days after infection, we could already see in one of the genotypes a really substantial reduction in feeding rate. So there were no visual signs of infection at this point. We had to actually keep them around in the lab for over a week after this to tell that they were infected, where it turns out we could have looked at their feeding rates and known whether they were infected or not. So this effect was starting much sooner than we had expected it would. And so the best fitting model for what's going on with feeding rate says that both host size and how infected it is influence the feeding rate. So the infected hosts increase their feeding rate as they get bigger, whereas with the infected hosts, it decreases as they get more and more spores in their body. Okay, and so the logic again is that the spores are building up in these hosts, the infected hosts. So the uninfected host is just increasing with size, but in the infected hosts, there are two things going on. So the infection is slowing their growth rate. They don't grow as much, and so then they're not going to be able to eat. So feeding rate in Daphnia is strongly related to their size. But then also there's something else about the spores that's depressing feeding beyond any effect on size. Okay, so then taking the lab results on feeding rate and looking at the theoretical implications of that feeding rate reduction. So we used a model that tracked the dynamics of susceptibles, infecteds, and spores in the environment and algae. And I will show you the equations on the next slide. My lab was like, no equations. And then I gave this talk at Florida, and Bob Holt was like, where are the equations? So I'm trying to keep everyone happy by having this on 2 slides. In the model, the infected hosts have a reduced feeding rate. And then we varied the algal carrying capacity and the sensitivity of spore yield to algal levels. And that allowed us to generate variation in epidemic size. Okay, so for the people in the audience who like math, this is the model. It's a fairly basic SI model with environmental transmission added on and algae added on. And again, we have this different feeding rate for the infected hosts, and then we generate variation in the size of the epidemics by varying the algal carrying capacity down here, and then by varying the sensitivity of spore yield to algal resource levels. Okay, so what we found is that epidemic size increases with increasing carrying capacity and increasing sensitivity of spore yield to algae. This is really boring. This is just showing us that the model does what we expected it to do. But I think it's useful to go through because it helps orient you to these figures. So carrying capacity is here, algal carrying capacity is here on the x-axis, and the infection prevalence is on the y-axis. And so as you move towards higher carrying capacities, you get more disease. And then the different colors in dashing indicate the sensitivity of spore yield to algae. And so as you go up, you get more disease. Okay, and so then as epidemic size increases, the population death rate increases and the population feeding rate decreases. Again, this is pretty boring. So this is a really virulent parasite, and we've told the model that infected hosts die at a higher rate. So it's kind of boring that the death rate increases, but again, this is sort of useful for where we're going. And now I've added on a line that shows if there was no disease in the system, what death rate would we expect? So you get this increase in disease as you get, or increase in death rate as you get more disease, and then a decrease in feeding rate as you get more disease. Again, because we've told the model that infected hosts feed less than uninfected hosts. Okay, so where things start to get more interesting, epidemics increase algal resources. So now we have algal resource density on the y-axis here. Again, our no disease case here. And as you get more disease, you get more algae. But where things get really interesting is if you look at what's going on with the Daphnia host density. So if you have low carrying capacity, you get fewer hosts during epidemics. You get a classic trophic cascade. You get more disease, you get fewer hosts and more algae. This is a classic trophic cascade. But the really interesting part is over here where you get a hydra effect. In this case, as disease increases, you get an increase in host density and you have your increase in algae. So everything is going up during the epidemics in this case. Okay, so this is a little confusing, right? You don't expect virulent diseases to be increasing the population size. But if we come back to this review by Peter Abrams, which is sort of the review of the Hydra effect, it talks about this third mechanism that can produce Hydra effects. So when the thing that's causing the mortality on your consumers leads to more prudent resource exploitation, then you can get a Hydra effect. And that's what's going on here. So the thing that's causing the increased mortality is the disease, and that's also causing reduced feeding rates, which is then more prudent resource consumption. Okay, so that was kind of interesting, not necessarily what we expected when we started this work, and so we wanted to see if we could see evidence of this happening in the field. And so for this, we used field data that we had from 15 lakes that are located near Bloomington that Spencer Hall's lab has sampled for a while now. And they were sampled every week from August through December. These data are from 2010. And they collected data on the proportion infected, host body size, which takes a lot of time to measure that, and then it's kind of mind-blowing to me that Spencer's lab could do this. They also measured the spore load in all of in a subset of the infected hosts to get the spore yields over time, and host density and algal density. And the body size and the spore load data are really important because that's what allows us to estimate what the feeding rate should be by pairing it with the lab experiment data where we figured out how feeding rate changes with body size and spore load. Okay, so we had all of the data then, and then we had to figure out what to do with it. So this shows for 2 lakes what the data look like. So the data for the algae is shown in green. The data for the host density is shown in blue. Again, we have this sampling over from September into December. And so one thing about plankton dynamics in lakes is that they are dynamic and they are very seasonal, and so things are very non-stationary in these lakes. So in Goodman Lake at the top, it had a really big epidemic, and both hosts and algae increased during the epidemic season. In Dogwood Lake, which did not have an epidemic, the host increased, but the algae decreased. And so what we did was we used the slopes as vectors, and then we calculated the cross product. So we could have the algal vector and the host density vector, and then we could calculate the cross product, and the area of that rectangle then tells us about how those two things were changing. And then in particular, if they both increased, it's positive. If one increased and one decreased, it's negative. And you might be thinking if they both decreased, it would also be positive, which is true but never happened in our data. So a positive means they were both increasing. Okay, so then if we look at the data, so the maximum prevalence of infection in the population is shown on the x-axis, and then again this cross product, this joint response of the hosts and the algae is shown on the y-axis. And when the epidemics were large, we saw that both hosts and algae increased during epidemics. So this ended up having a positive correlation, was just sort of like the icing on the cake. Really what we were just looking for was whether we had these positive cross products with larger epidemics, and we definitely saw that. And so again, this is evidence of a Hydra effect. And then we also, because we had all the data on host body size and on spore yield, we could calculate how much feeding rate should have been depressed. And again, we see this very strong positive relationship there where when there was greater feeding rate depression, this cross product was more positive. Okay, so I realize that this story has a lot of parts and there's a lot going on. So to summarize it, if you have a large epidemic, you get a decrease in feeding rate because you have a lot of infected hosts and the infected hosts eat less. And that leads to an increase in the algae, and that actually feeds back to lead to a larger epidemic because then you get a higher spore yield, all else being equal when you have more algae around. And then your increase in algae leads to an increase in reproduction, and this is especially happening in the uninfected hosts. And we see signatures of this in the data, that when there's a big disease outbreak of one of these virulent parasites, the uninfected hosts, their fecundity actually increases. So it's freeing up resources in these lakes. And then that can actually more than offset the loss from the disease, and you can end up with an increase in host density during these large epidemics. And so the take-home message then is that trait-mediated indirect effects of parasites can drive trophic cascades and can actually increase host density. And one reason why I think this is interesting is that parasites, like by definition, they harm their hosts. And we think of these parasite epidemics as things that harm host populations. But we actually don't generally see populations being just obliterated by parasites, even though there are a lot of virulent parasites out there. And so clearly there are some mechanisms going on that are allowing host populations to persist, and sometimes with very little effect on their population dynamics in the presence of these parasites. And so it's interesting that in some cases you might actually be able to see an increase in host density during large epidemics. So a really obvious question then is, how general is this phenomenon? Right, that's sort of like to me, the most obvious first question. And so we wanted to see if this held for this other common parasite that we have. And this is work that was led by my former postdoc, Catherine Searle, who's now at Purdue University. And she did this with the bacterial parasite that I talked about before. And so what we wanted to know is whether feeding rate is also reduced in infections with this bacterial parasite. And it is also, again, it's strongly reduced, and it's reduced early in infection. So here, there's, again, a wide range of time since infection, and there's the feeding rate on the y-axis. And these infected hosts in red are clearly below the uninfected hosts in blue. This is particularly notable in this system because the infected hosts in this case are actually larger than the uninfected hosts, because this parasite induces gigantism. But the effect on the feeding rate is so big that it offsets that increase in body size. And so this makes us think that maybe trait-mediated effects of parasites on host feeding rate might be common. The only two systems we've looked for it in, we've found it. So that seems kind of interesting. And so then to me, this is this classic picture from an experiment by David Schindler, where they added phosphorus to one side of this lake and not to the other. And I think it's an interesting thought experiment to think about whether we could generate a similar effect if we added a whole bunch of parasites to one side of the lake and not to the other. I definitely think that something that's important is that the host is the dominant grazer in these lakes. So if this was like a copepod that's a really small fraction of the population, we wouldn't expect to see these ecosystem level effects of disease. But because the host is a really key player in these food webs, then I think we can see these effects. I also think it's really interesting then to think about how the Daphnia parasite interactions are influencing resources. And then what I told you about last night is how resources are influencing the Daphnia parasite interactions. And so just very briefly for people who weren't there last night, so work by my student Crystal Sanchez found that these different diets of phytoplankton that you feed can strongly influence whether the fungal parasite is even able to infect in the first place. Interestingly, that's not true for the bacterial parasite. So there's a parasite identity effect here that's important too. And so if we come back to this food web that I looked at before, so the lakes are getting browner, as I mentioned, but they're also getting greener. That's another part of global change. And so we're not only changing the brownness of the lakes, what I talked about in the first half, but we're changing the greenness, and now we're starting to realize there are all sorts of links between the disease and the primary productivity. And I think it will be really interesting to try to figure out the net effects of global change in this system. And so we also, in this talk, I've mostly been talking about have one host and at most two parasites. But another complexity that we need to consider is that there are multiple hosts, right? So we have just one Daphnia there. There are actually, our lakes range from about 2 to 6 or 7 daphnid hosts in them. And then we have more than just these two parasites. And so I didn't have time to talk about it, but some new work that I'm doing with Mike Cortez is looking at how, when you have multiple hosts and multiple pathogens in a system, how does the competition between the hosts and the competition between the parasites end up influencing these dynamics? And this is important, I think, not only in our Daphnia systems, but also much more broadly. So most of the emerging infectious diseases in people come from, they're mostly multi-host diseases. They're coming from an animal into humans. And then, as I know all too well from daycare, there are often multiple pathogens circulating within a population at a single time. And so we have to realize that there are going to be interactions between the hosts and the parasites, and those are probably really important. Okay, and so with that then, I just want to end by summarizing the two parts of the talk. In the first part, I talked about how light drives parasite dynamics in our Daphnia parasite system, and our data suggests that a browner world might be a sicker world. And then in the second part, I talked about how disease can increase host density, which is something known as a Hydra effect. And this is happening because of a reduction in feeding rate that increases the primary productivity that then allows the Daphnia to sort of offset the loss that occurs due to the disease. And with that, I'll thank you for your attention. Yes.
You alluded to this right at the very end of the talk, and I missed your talk yesterday, so maybe you
Sure.
covered some of this. But all the browner world stuff, I keep thinking about a greener world because we continue to have so many problems with eutrophication. And so the first question is about the browner world. Are these shallow systems? Because you said that this is mixed up DOM that's coming up. And so are these relatively shallow and therefore storm effects make them browner?
No, so these are all stratified dimictic lakes. And so the brownness is coming from, there's just more coming in throughout the year, and then it can get mixed. And it definitely stratifies, but the lakes are still pretty brown throughout the year.
Playing with the greenness of it, not the
Yeah.
brownness of it. It seems to me that if you've got Daphnia grazing algae, and hence making it a less green world, you're going to increase the transparency. You're going to increase radiation into your lake, and that's going to be to the detriment of the parasites. And so you're going to have a positive feedback on the Daphnia. Have you guys been looking into that?
I really want to, and so I'm like masquerading as an ecosystem ecologist. I'm not actually one, but Jake Allgaier was just hired in my department, and he's a really good aquatic ecosystem ecologist. And we've been talking about doing some work where
it seems like such an obvious.
it really
also, the
does.
linkages with Brooks and Dodson's kind of top-down sorts of things as it pertains to Daphnia control and implications of that.
Yeah, and there's a grad student in another lab who's looking at cyanobacterial blooms. And so in my dream world, we're going to measure the nutrient dynamics, we're going to measure the phytoplankton dynamics, and we'll measure the Daphnia and disease dynamics. And then
That'd be really interesting.
I would really love that. Yeah.
Thanks.
Yep.
If your Daphnia reacts differently based on their genotype, and you're working in lakes that are in Michigan and Indiana and Pennsylvania, are the genotypes really constant and similar across those areas, or how do you control for the genotypes when you're doing the field portions?
Yeah, that's a great question. So the genotypes definitely differ, but there's huge variation within each population. And so when we look at basically any of the traits we're interested in, we see a lot of variation within each population. And the mean values of the traits do differ somewhat, and that I've actually for a while I worked on how these epidemics were selecting on traits like resistance and feeding rate. But it seems the main effects actually seem to be variation within lakes or over time, as their selection. And when we look at the phenotypes, we don't actually see big differences between the populations. We've actually also been doing that work on the parasites and looking at their genetic variation. So something that's really weird that we can't explain is we find very little genetic variation in the fungal parasite. And this makes no sense. And I've spoken to a lot of people about this, and everyone thinks it makes no sense. But we've actually looked there. The genotypes are actually very similar between Michigan and Indiana. And again, we're not really sure what's going on. It's a little weird. Yes?
Just curious. You suggest that a browner world might be a sicker world. But there are large parts of the world where the water is much browner than your brown lakes are now and have been as long as they've existed. any idea what might be going on in those systems?
No, because people haven't looked at the parasites very much in these. And one thing that's one thing that's interesting is in ponds, we don't see a lot of disease, at least in ponds in Michigan when we look. But you get different host species there. And it seems like the host species differ greatly in their susceptibility to the different parasites. So we haven't, so we definitely have some ponds that are pretty brown and they don't have much disease, but they also have different hosts that we can't infect in the lab either. And then generally we don't have good data globally yet on what's going on with disease. There was someone in New Zealand who recently assembled, did the best job of assembling a database, but mostly people don't look at Daphnia parasites, and so we mostly don't know what's going on. Yes?
With respect to the fungal parasite,
Uh-huh.
the browner world wasn't as sick a world, even though the fungus was sensitive to light. So I'm just curious at, well, two questions. Why do you think that is? And is that a big deal, or is it like, well, it could be anything?
Yeah, so the fungus, I think it is a little confusing because we do see an effect, the effect that we would have expected in terms of the timing of the epidemics, but we don't see one in terms of looking across the lakes in terms of the epidemic size. And so one of the things that might be the case is that some of the other food web factors are swamping any brownness effect. So we know especially that invertebrate predators are really, really important. So Chaeabris, midge larvae, they're sloppy feeders. And so they shred these infected Daphnia out in the water column where they then do a really good job of spreading disease. And then conversely, fish are really important at reducing disease because they feed selectively on the infected hosts. And then that should interact with water clarity also. So,
But was, on that reason right there, was there a difference between the bacterial host and the fungal host? Or, I mean, are they,
in terms of what?
well, you're explaining
Oh, is
why
there?
the sloppy
Yeah.
feeders might
Yeah.
influence the fungal host. but
Yeah.
Did the epidemics happen in the same lakes?
Yeah, well, we see much bigger epidemics of the fungal parasite than of the bacterial parasite. And so if there's something where, there are thresholds, right? And so for the bacterium, it might be that it's sort of close to this threshold where it just can't take off. But for the fungus, we're not sure why, but maybe these food web factors are allowing it to be well over the threshold, and that's swamping the effect that we might see from the brownness. But we don't know. We actually tried to do, Clara made this heroic effort to do a whole water column enclosure experiment where she factorially manipulated light DOC levels and then also UV penetration. And it was a really, really hot summer and everything died in every bag because it was like 30-something degrees in the Epilinnian, and it was very sad. Yes?
So you've just sort of been saying light, and then you sort of say UV. But when the color changes, the light penetration shifts. And so I'm just curious, if you've actually broken apart what colors we're really talking about, what wavelengths we're talking about, and is that part of the story? Yeah,
so I think it might be part of the story, and so it's not just UV. It's photosynthetically active radiation also, and so the blue light especially is damaging them. Okay. And so we've done work with, the light work is all in collaboration with Craig Williamson, and we've used his Phototron where you can really carefully measure all of this in the lab. And it's definitely more than just UV. And we've also then used his BIC to quantify the whole spectrum in all of our lakes. It's hard though to do, and so we've thought about should we look at the amount in certain wavelengths, but then there's also some evidence that it might be the ratios that matters, and we don't have enough. It feels like a fishing expedition once we started heading down that road because we didn't have clear enough hypotheses about what we should be looking for. All right. Thank you.